Hi, everybody. Today we are going to talk about transposons and horizontal gene transfer. This is going to go along with the viruses. Now, you may recognize the term horizontal gene transfer because we've already talked about it in terms of bacteria. For example, transduction. This bacteriophage is infecting a bacteria, and therefore it might change its genome. That was transduction. That's a type of horizontal gene transfer. Conjugation, you see that here, sending one plasmid over from one bacteria to the next. That's another type of horizontal gene transfer. And then also taking in free DNA was a type of horizontal gene transfer. Now we're going to talk about transposons today, which will go with virus, and this is another way of mixing up genomes. So transposons are really incredible, strange elements known as jumping genes. And they are long, repetitive stretches of DNA sequences that were identified only about 50 years ago by a geneticist named Barbara McClintock in New York. Biologists were initially skeptical of her jumping gene discovery, which as we talk about what it is, we'll see why. It's pretty incredible. However, eventually it did become apparent that these transposons jump. And not only that, they're not rare. They're very common. We find them in every type of organism, and we actually find quite a few of them. Transposons resemble remnants of ancient viruses, which gives us a hint as to where they may have come from. It could be remnants of viruses that have inserted themselves into their host DNA. When active, transposons copy themselves and then jump around, inserting themselves somewhere else in the genome. In fact, they can insert themselves right in the middle of a gene, therefore interrupting and destroying the gene. When a transposon is activated, it can insert itself within a critical gene, which can cause serious problems. It can cause infertility. It can cause a genetic disease, right? We don't need or we don't want our genes interrupted. To combat this ever-present threat from within, the cell has devised stringent mechanisms to maintain tight control over transposon activities, largely through epigenetics. Epigenetics, remember, like methylating areas of the genome can turn a gene off. So we can, for example, methylate areas that the cell believes might be a transposon to turn that section of DNA off. Regions, so the second bullet point is important. Regions of the genome that are rich in transposons typically have inactivating signals, which silence the transposons. Also, microRNAs, remember RNAi? Those microRNAs that can chop up and block pieces of the genetic code might actually target transposons when they are activated, thus stopping the transposons from operating. There's a picture of Barbara McClintock there, and here is how her discovery of transposons worked. Here's the transposon, here is the target site. The transposon is going to copy itself and move over here. The bottom one is the copying itself. The top one is simply cutting this transposon out and pasting it into the target site. So there's really two methods. It's either copying, like you see on the bottom, the transposon simply copied itself and moved it to the target site, or the transposon is cut out and then the whole thing simply moves to the target site, interrupting that DNA sequence. Cut and paste or copy and paste. All right, now transposons contribute to spontaneous mutation, they contribute to genetic rearrangements, and they contribute to genetic diversity. Transposable elements can be DNA or RNA. They can seem to come from a viral place or a non-viral place, and they, as we talked about before, they can be replicative or they can be excision-based. So here's our transposon. It has copied itself and inserted itself into this piece of DNA. Now this, in this diagram, this is actually showing the chromosome of a bacteria, and this is a plasmid. So in this example, we are transposing from the chromosome to a plasmid. So bacterial transposons move within and between chromosomes and plasmids. And if we combine that with the information we already know about plasmids, the transposon that's now present in the plasmid can move to another bacteria through the process of conjugation. Okay, we're going to skip this. So, what are the implications of this? When Darwin proposed his ideas about how new species arrived, he produced a metaphor that we still adhere to today to explain the branching patterns of speciation, the tree of life, right, branching. But Darwin assumed sort of vertical gene transfer, that things always got new genetics as they reproduced. And now we know that all of this horizontal gene transfer is actually really going to complicate the tree of life. Notice here all of the horizontal elements from one branch to the next. We see things like conjugation, transformation, transduction, moving plastids like a chloroplast to mitochondria, and transposons make the tree of life a lot more complicated. The method of gene transfer that would produce a genuine circle or ring is horizontal gene transfer, in which two organisms fuse genomes to produce a new organism. An example of this, the endosymbiotic theory. Two organisms came together and they have made something new, a eukaryote. So the tree of life is perhaps too simplistic, that it is a little bit more complicated, and some scientists have hypothesized that it is even really more of a ring. All right, and that is all for now. If you have any questions, let me know.